from Prakriti and my conversation. So we have a diverse background of people and from the different domains. So I thought my talk should be more of a application oriented, not into the too deep of the coding, as well as too not too much into the drug designing. So I changed my presentation to the very simple and case studies and basically the concepts. So here is my outline of the talk. So I'll start with the introduction to NLP. So my brief talk starts with the drug discovery workflows and followed by the NLP use cases where natural language process can be applied in the drug designing and where currently I'm applying. And one of my strongest zone in NLP is the generative chemistry and transfer learning. And I'll show you some of the success stories of generative chemistry. And then followed by, I pretty much day-to-day basis, I use LSTM models for my drug discovery project. So I'll start with a simple introduction to the LSTM models and how the molecular representation works. I have a simple Python code demonstration also if you're interested, and then followed by how LSTMs works. And followed by case studies, I actually planned, there was a genetic chemistry success stories, and we have successfully designed molecules using AI and synthesized and tested, and we achieved nanomolar potent compounds. Everything is from AI. So, but unfortunately, publication still yet to be uploaded in Chem Archive. So probably next week or by next week, it will be available in Chem Archive. So I'm really sorry I'm not able to share those details currently, the case studies, which I'm
Sunil, one
planning to
minute.
discuss with you on H.8.
Hi, Sunil. Sorry to disturb you. We are in your slide outline of my talk. So is it in your present slide? Am I right?
Yes.
Okay. Can you move slide? Yes. I just want to check it's moving is visible. Money is internet connection is okay or not. Okay, Sunil. Yeah, please.
Okay. Can you see a full screen or regular
Yeah, I
presentation?
guess we can see you in a full screen.
Okay. So thank you, Prakriti. Right. So let's start with a small introduction to the drug discovery workflow. And most of you already aware of this the workflow. Maybe you are already know better than me. So the drug discovery cycle starts from the basic discovery, understanding of the target, and start with the hit identification and optimization, lead discovery and lead optimization, and identifying the best candidate for the further in vivo studies, and followed by one best molecule goes to the clinical trials. And based on the clinical trials, if everything goes well and data is promising, so it goes to the FDA review. And everything it takes almost like a 10 years of span and almost like a 2.6 billion cost. So you can imagine if any molecule that fails in a phase one and phase two, almost we are going to lose $1.5 billion and 9 years of effort. And drug discovery, is not that easy, and success rate is very, very low. And, you know, but still, it's a long way to go. Right. Now, if I see the breakdown of the entire cost, uh, as I mentioned, the drug discovery starts with the basic biology and target to hit and hit to lead and lead discovery, lead optimization. So basically, I play key role in the molecule designing from target to hit and hit to lead and lead out. So only these four pages that cost almost like a 800 $800 million dollars, and it takes 5.5 years, and almost the failure rate is 69 to 85%, not success rate. It's a failure rate is 69 to 85%. And if you want to reduce the cost and if you want to reduce the time that takes to design a molecule and to launch into the clinical trials, so this is the best page where we can reduce the timelines and cost and as well as the failure ratio. So how? Is there any examples? Let's see some of this page. Okay. Now coming to the hit identification. So if anyone wants to start with the molecular designing, so we'll always start with the, okay, let's take a kinase, let's take a GPCR target, which are important for the CNS or cancer, then understand the protein structure or understand the clinical candidate already exists on the target of our interest. Then start with exploring a new molecular design that we call it as a chemical space exploration. So in this chemical space exploration, all we are trying to take a millions or billions of database and trying to find the molecule of our choice out of billions. And what is our requirement is the desired properties. It should be drug-like, soluble, and less toxic, and the compound should be active on the target of our interest. So there are so many methods that exist uh, that are already we have been using successfully in the industry. They call it as a molecular dynamics, quantum chemistry, molecular dynamics and virtual screening, docking studies, where we use these techniques to filter the billions of compounds and try to identify the top 100 or top 50 molecules. And in order to do that, there is a pros and cons of each approach. So when I see the virtual screening approach, which we call it the inverse approach, we'll take a billions of data set and we'll try to screen against the target of our interest, simply the docking, and we'll try to apply multiple filters. Like we don't want toxic groups and we don't want the reactive groups, and followed by we want a certain molecule, certain features. I don't want certain substructures like a paints. So I can apply n number of filters I want finally, so that I can reduce the billions to maybe 50 compounds, where I can pick those 50 compounds and purchase and screen it against the target. And in order to do that, you need a huge competition power. And if you want to screen billions of compounds, you definitely need a high-end cluster. And it takes minimum 1 week to 1 month time, right? And again, success rate is always depend on the choice of your docking engine, the choice of the filters, and choice of the protein structure. There are n number of points where you can go wrong as well. Right. Okay, now comes to the picture. So we discussed about a traditional approach or inverse approach. Okay, now coming to the generative chemistry. You must have heard about this term called generative AI. So, ChatGPT. Now everyone is aware of what's going on in AI because of ChatGPT. So similar to generative AI, so generative AI is also applicable to the drug discovery, that we call it as a generative chemistry or de novo drug designing. The inside the architecture is either variational encoders, or LSTM, or GANs, or reinforcement learning methods, or transformers. So all are nothing but of different architectures and trying to design a molecule of our interest.
No, I have.
Sorry?
Sorry, Suneel. Sorry, please carry on. Okay, sorry.
Okay, right. Okay, now in the natural language processing, is generative chemistry the only thing that I can use? Is there any additional application? No. In natural language processing, we can actually use for multiple purposes. We can try to understand the structure activity relationships and trying to do the patent analysis. And we can do the chem genomics, and where we can extract a relationship between the targets and molecules, and simply the polypharmacology, and enhancing the bioassociative identification simply when we're trying to do the patent busting or new IP space. So we can use NLP like a focused deep learning based scaffolding tool, and followed by the data-driven molecular designing. That's alternatively we can call it as generative chemistry or de novo designing. And the next one is predictive modeling for ADMET or solubility properties, where we use the NLP techniques to use, trying to extract the information and trying to predict whether the given molecule is having ideal properties or not. And not only that, we can use the reinforcement learning technique to select the best molecule and try to avoid the noise from this. Okay, so far any questions? Shall I continue? Okay, so I assume it's good.
Please continue.
Right. Thank you. Okay, so you can coming to the NLP, you can generative chemistry or AI. So the first thing that comes to the mind, the problem that what we discussed, drug discovery has a high failure rate. It takes a time and it takes money. And we discussed about NLP role in drug design and how we can actually address the ultimately the failure rate in the drug discovery and timelines. I have a very well-known success stories that can address all these questions. The first one is in silico medicine. You might have already know this success story. It's a way back in 2020 during the COVID time, where you must have seen these articles like this startup identified a drug within 21 days. If you go back to the 4 slides back, we discussed about it takes 10 years, it takes $2.4 billion. So here this guy is saying 21 days. Actually, it's a drug candidate, so which is a potential to be launched into the clinical trials, or maybe the ideal candidate for the assets and to go into the next stage of optimization. So here, the in silico medicine is one of the well-known startup in artificial intelligence, where they use a generative AI, where simply they call it as a generative tensorial reinforcement learning algorithm, general. So they used this algorithm, they trained this algorithm on their kinase data set and the targets of their interest. And they generated ideas from the generative AI. And based on the generated ideas, they further processed and docked those compounds and try to look for the similars in the commercial compounds. And they synthesized and they tested very, very less number of compounds. And they identified not only the nanomolar potent compound, they also came up with a great profile in the in vitro in vivo assays. And that molecule has a good drug-like properties that can be translated into preclinical candidate. So I think overall they tested 45 compounds, if I'm not wrong, and within a span of 46 days, where they completed designing, testing, synthesis, and in vivo, in vitro, all the studies. And it took hardly less than 2 months, whereas a traditional process, it takes 5.5 years and takes $800 million. Here, they spent hardly less than $1 million. We can see the success. Right. So the next one, Accenture success story. This is the first success story where they come up with on the adenosine A2A receptor. And within a span of 9 months, they identified a clinical candidate by just testing only 160 compounds. And currently, this compound in clinical trials. So you can see the timelines. They have a clinical candidate within a span of 9 months. And the technique that we used, simply the AI-based molecule. And another clinical candidate is a well-known company, Schrödinger. So they used their strongest power, physics-based machine learning models, and they synthesized hardly 109 compounds, and they come up with a clinical candidate within a span of 10 months, and against the MALT1 inhibitor, against a blood cancer drug. So these three success stories actually giving us a clue that yes, there might be a solution where we can actually improve the success stories, and we can we can also address timelines and costing and everything. But again, we can't apply this to every drug discovery story and launch a drug within a span of a few years. So there is always pros and cons, and but it's one of the alternate solution, I can say. Right. There are many more success stories like this. There are climbing like a powerful antibiotics were discovered using AI. Similarly, there are powerful cancer molecule was discovered by MIT. So there are many success stories that we have been hearing about it. So that's all about introduction part, and let's move on to the next one. It's about the small introduction to the NLP. Okay, so before I move into the NLP, like one basic question that comes to the mind is, okay, why I have a chemical structure. So how, the machine learning algorithms, how these tools read the molecule and use that molecular information for the either deep learning model or machine learning model as the input. So for a given molecule, there are multiple types of representation is possible. One is SMILES, which is a simplified molecular input line entry system. So that widely used format for representing chemical structures, where we call it as a 1D string, which is a human readable format. So we can simply, take the entire molecular information into 1D and convert that information into fingerprint and convert them into bits and use that information into the either ML or deep learning models. And the next one is 3D representation. So which is for either 3D coordinates or electrostatics, where we call it as electrostatics and shape. So when we consider, there are pros and cons and again, everything. When you consider smile string, which is a very fast to train a model, but when you consider 3D coordinates or electrostatic or shape, you are considering the entire molecular features in terms of the shape, in terms of orientation, in terms of molecular graph. So there is a lot of information can be extracted from a single molecule. Now imagine you are trying billions of compounds, so it need a lot of computation power and time as well. So similarly, the fragment-based approach, electrostatic-based approach. So all these are time-consuming, but it will add a lot of strength to the model because whereas a simple 1D smile string, it simply represents molecular structure. That's it. It won't capture the complete essence features of the molecule. Right. Okay. Now coming to the NLP. So where the NLP is basically is the text-based approaches, where we have alternate methods called graph, or we have alternate methods called shape, or either 3D, followed by the transform-based approaches. But in the NLP, we simply convert the entire molecule into the smile string, which is nothing but of a text, and we use those to train the models called either LSTM or GRU or transformers. Right. Okay. So before I start, why AI is the famous AI is more of widely used either ML. So because of the one point that here I want to stress is it's open source. And because of the open source, a lot of active research is going on, a lot of validation is going on, whereas the commercial tools, they have a limited accessibility to only certain people who will take the license to their software. Whereas because of these open source tools, they are well validated, well explored, and the complete information exists available on the internet. So if you see the basic machine learning workflow, so we'll simply try to take a data set of our interest, whether it can be target or it can be ADMET or it can be docking related information. Simply then start with the data analysis and data processing, then feature extraction, then property analysis, understand the statistics and property distributions, and followed by ML or DL algorithm, then followed by validation, ROC curve, and other matrix. Then once all the statistics are good, and apply those ML model on the virtual database to rank them or filter them. So in order to do the entire activity, so we have certain libraries which can open source, and there are some commercial libraries as well, which will help you to perform this task. So here, there are certain commercial tools like Chemaxon and BioVR, Schrodinger, and Pipeline Pilot, where they are commercial. Otherwise, the rest of them is open source. Right. Now, that is what the packages that we need in order to start with NLP. either ML, simple ML modeling. Now, coming back to the generative AI in drug discovery, the first model that is proposed way back in 2017, and there are n number of based on input or based on the approach, they are classified into encoder or decoder based models. They simply called as VAE models, and RNN, GANs, flow-based, conditional, graph-based methods, and there are huge number of diverse models has been proposed, and basically all these models have again pros and cons. If you are planning to take VAE models, they are quite fast, and sometimes it gives a very surprising results or out of box results. And coming to the RNN, it's a well explored, widely used technique. Sometimes when you wanted more ideas, it ended up generating duplicates. It ended up generating non-relevant structures. And graph, yes, whereas a graph, we are taking the molecule as a graph instead of a smile string. But computationally, it takes a huge time to generate and train the model. Okay, now it's a simple statistics. How generative chemistry models have been published over from 2017 to 2021. I don't have a recent statistics. If you see majorly, like there are techniques, as I mentioned, like RNN or encoders or GANs or variational encoders or RNN LSTM. So RNN LSTM is the most famous or widely used in the publications or maybe in the reviews compared to the other techniques. So, and it's very easy to implement as well. And yeah, so whenever you can customize your models within a short span of time. And RNNs are nothing but of a recurrent neural network models, simply a type of deep learning model that can use to generate text AI. A very simple example is, WhatsApp. When you use iPhone or Android phone, when you're sending SMS or when you're sending a WhatsApp reply, you can actually see what is the next probable character. When you say hi, so the next probable character is where, how, and something like that. So similarly, RNNs in a similar how the text generation or text prediction is coming up. So similarly, RNNs are applied to the smiles as well, where it try to understand the existing chemical space and their smiles distribution. And when you ask RNN to generate a molecules of target of your interest, so it try to you know build a molecule by character, like a carbon. Okay, what's the next probable uh, atom? So nitrogen. Next probable atom can be carbon because it learned from the distributions and it learned from your fine-tuning data of your target data, and based on the learnings, it will memorize and it degenerate molecules by combining by calculating the probability of distributions. Right. So it's a very simple model. I took an example from the reinvent dataset, reinvent model. So where here the general prior is nothing but of it, the basic RNN model that is trained on the Campbell dataset, or e-molecule database or Jing database. Whereas a Campbell is nothing but of biological active database, where they have around, I think, 4 million compounds, and followed by they have a biological activity, and they are curated manually from the journals. So it means that all the molecules that are present in the Campbell, they are already synthesized, they are active against some X target. So we are using those Campbell database to train our RNN model, our LSTM models, our transformers, so that model will learn the actually synthesizable compound database, and they are biological active as well. And then we can ask random sampling. Okay, once the model is trained, you can ask a system to, okay, now generate a molecules randomly. So it starts sampling the molecules from, simply by carbon, next character nitrogen, install, slowly start building the molecules. So then once you have a random molecules, then you can actually use some machine learning model, or you can use the docking studies, or you can use some scoring system to process the generated compounds and shortlist the best ones for you for your next iteration of census. Okay, so let's make it more complex or more ideal approach, what we follow in the industry. So first one is general prayer, which is a biological active space, which we are trying RNN from the chemical data. And coming to the small data set, imagine you are working on a GPCR, imagine you are working on a kinases. So instead of a biological active space, you want to provide the model to, okay, so I know it's a biological active space. They might be ion channel inhibitor, GPCR inhibitors, or kinase inhibitor, but I want to give attention to the my small data set where it's a kinase specific, a GPCR specific data set. And we'll use the transfer learning approach where we'll build the focus prior. So that's called a molecule. The model is trained on the target of our interest or the interested family database, and then sample. So whereas initially it's a random sampling, now when you train on the focus target or family data set, you'll have a molecules more refined and more meaningful data sets. And then once you generate, you apply all the filters that you are looking for and narrow it down to maybe hundreds or 200s so that you can handle this number. Okay, so this is a much more complex one, but ideally I can say this is a workflow that we follow for the most of the people will follow for the molecular design. And the next one is simply where we use the reinforcement learning. It's the same thing. General prior plus a small data set. It can be target data set or it can be family data set. And then we use the transfer learning where we have both general prior and focus prior. And here we'll use the reinforcement learning where we tell the RL to design or process or filter the molecules that are having certain characteristics, either properties or either you want to consider some substructure, you want to filter some substructures. Yes, you can tell the agent where agent takes care of your data set processing and filtrations as well. And then you try to generate the molecules within the given conditions. Like imagine you want to have a molecules from 300 to 400 molecular weight. So whenever the molecule generation is happening, so this RL will take care, it calibrates a molecular weight for each compound. And if it is any compound has a 401 molecular weight, it discards. Then it only saves the molecule that satisfy the filter provided by the user. Then at the end, you can only see choice of your data set rather than all the combinations. But I have a pros and cons for this when I use the RL, because when you use the RL reinforcement learning where you want some substructure or you want to discard some substructure, you want to assign some property thresholds. So there is a more chance that you might miss some ideas. You might miss some, interesting candidates, which are ultimately that's what our interest. So you can apply RL whenever you have a requirement to, okay, now I know the ideas, but I want this rule to be satisfied so that I can save the time in the later step. Right. Okay, so in general, how do you assess the genetic chemistry data sets? Okay, you used any workflow, either random sampling or target-based sampling or using RL, it generated a molecule. So how can you say the generated molecules are valid, satisfying my requirements? So there are some benchmarks, there are some data set, there are already known GitHub repos, they call it as a Moses, where they use a benchmark the data set, where they assess the smiles quality and novelty, but ultimately it's a user has to decide they are meaningful, good to be considered for the synthesis or not. This benchmarking will simply tell you the generated smiles are valid or not. They are unique compared to this given data set, are diverse enough or not, novelty, whether they share some similarity with uh, the target of your interest or not, or finally synthetic feasibility. So when you use the benchmarking, it automatically calculate everything and will give you the statistics. But this statistics is only to assess the given smiles, but it won't tell you about the targets. Given molecules are very good binders. No, it won't disclose that information. Right. So these are some success stories. I was supposed to discuss my success stories here, but because of publication didn't move to the next level, so I thought I can discuss about published success stories. So there are some, as I mentioned, the genetic chemistry was initially proposed in 2017, and now they're aggressively using it. So there are different, there is a phenomena, there is a misunderstanding. Always like we consider deep learning algorithm, we need a huge data set to train, or maybe genetic chemistry generate invalid smiles, or they are synthetically not feasible, the ideas won't be that great. What are the filters I need to apply in order to achieve the better compounds? So to address all these questions, I've made a comparison here. You know, out of these three success stories, which are published by the different research groups from the University Oxford or juries or in silico medicine, you can see the data set. In the first case, they trained 25,000 data set as a focused prior, and whereas second case is only 25 compounds, and third case 30,000 compounds. So it worked. And finally, they generated 10,000 compounds here, 1,000 with a substructure. So the author wants to have a molecules with a carboxyl group as for sure. And the next one, they generated 30,000 compounds by using AI. And typical filters are solubility filter, synthetic assessment filter, and followed by the dot score cutoff or Metcalfe filters clusters. And is there any success story? Yes. So they tested 5 compounds in the study, and the 2 compounds are micromolar potent. And here they tested 5 compounds, and 1 compound is nanomolar potent. And in the third case study, they tested 50 compounds, 7 compounds are active. And similarly, there is more. So there is a success from the generative chemistry, but only one point is like, when you use the reinforcement learning, when you use the general LSTM approaches, so we need to, one should make sure that, there might be ideas that are noise, there are files, they are invalid. You can ignore those and process those filters and assess the molecule, whether they're synthetically feasible or not. There are a lot of articles, SS score, or you can use a axis or Sci-Finder to assess whether they're synthetically feasible or not. Then you can take it to the next level. Right. Now coming to the limitation, the basically, one limitation is, imagine I have a target and there is only 2 compounds reported so far. So will genetic chemistry works? No. So when you have a low data regime, so where deep learning algorithms fail to read and understand the distribution and generate a new molecule. So there are some case studies, even with the 10 compounds, 20 compounds, or 30 known compounds, yes, it worked. But with a very less number, you can expect a high noise rather than meaningful ideas. And there are some approaches, they call it a genetic algorithm-based derived genetic chemistry models. They call it as selfies. They are much more meaningful. Even with a single molecule, you can mutate and generate more ideas. And lack of, second point is lack of unified evolution protocol. Although they say benchmarking protocols, they simply assess the given molecule, they are valid or invalid, diversity or similarity calibration. They don't know how to assess the given data set. And there are sometimes we observe like in a given data set, out of distribution, like the molecule they don't find, they're not meaningless compounds. So we can find, bulky compounds or too flexible compounds sometimes. So there's no filters or to reduce that noise. It's all depend on our training set and lack of interpretability. So like in machine learning model, we have a lot of, parameters, a lot of points that we can take care, make sure the model is great or model is good or model is bad. Whereas in genetic chemistry, so we don't know uh, till directly we can say it's a good model or bad model to generate ideas. So these are the limitations, and still there is a lot of work is going on. And I'm fortunate enough to be part of it and working on the AI, especially in the ComChem side. So, and I'm happy to reconnect with you and share my success stories once it is published. And that's all for I plan for today. If you have any questions, and I'm happy to discuss and share.
Thank you, Sunil, for a nice talk, and this is very nice talk, and it's very clear. So I will definitely hand over the mic to the audience. But before that, I would like to tell you that it is really, I feel really good that with the 25 molecules using the LSTM, the success was more than 80%, right? In that paper, they have 25 molecules, and at last, out of five,
Yeah,
four is success. And that gives me a really, because
right,
nowadays, if I go in some lab, they don't have the much more data. They have 40 compound or maybe 50
right,
compound. So always I think that is
right,
it possible to apply in generative model or not? So you give me the confidence that it's possible. You give me the confidence today. And Shruti, I just want to ask
right.
one thing that there
Thank
is
you.
lot of different generative models like GAN, transformer, VAI, or flow, or regression most probably. So do you tell me that which one may you have used lot of generative AI in your career? So do you find that which one is best with irrespective of the
No,
data set?
so it's like you know, if you ask me the same question in a alternate way, like there are, various iPhone, cell phone models are there, like iPhone, Samsung, and other
Yes,
brands. So, everyone has a different choice. So it's all depend on the user.
true.
Each method has
Okay.
pros and cons, as I mentioned multiple times. The
Yeah.
graph is very good, whereas it works in many cases because it captures the
Mhm.
2D orientation and 2D
Yes, true.
geometrical information, but it's a time consuming. Are you okay with that? Yes, graph is very good because
Okay.
whereas LSTM, it uses smiles information, whereas a graph, it uses a 2D structure of the molecule.
Yes,
So
yes.
it's all like when you make it more complex, model can learn more and it can actually train and understand better and it can generate more meaningful results. But again, it's all depend on the user perspective. Are you okay to train the model for days and wait for the results for a days? Yes. Whereas the LSTM,
Uh,
it takes only 3 hours to train a model. Sorry,
True.
train a model, it takes 8 hours
Yes,
and generate a results, it takes only 2 hours.
yes, yes. True, true, Sunil. Yeah,
Yeah.
thank you, Sunil. So now, audience, you
Thank
can
you.
open your mic and directly you can ask question to our speaker, or you can raise also your hand. Then you can, if you want to type in chat box, you can. If yeah, Pooja Chandan, please open your mic and ask the questions to sir. Hi, Pooja Chandan. Pooja, your hand is raised. Your mic is mute. Could you please unmute your mic, Pooja?
Okay, I will ask.
Yeah. Yes, yes, sir. Please.
So now lot of single cell DNA sequencing, lot of sequencing is happening on human cells. So how it is getting, these two areas now, the sequencing and drug discovery. Okay. Are there is any connection?
Right, sir. So honestly, I'm not a right person because my domain expertise is computational chemistry and AI. But all I can say, like technology is advancing a lot, and which will ultimately helping us to identify, the more druggable targets and more meaningful targets to design the molecules. That's what I can say. I can't go more into the deeper into the subject because it's slightly away from my domain.
Okay, Dr. Suneel, I would like to introduce you. Now we are talking with our dean, Dr. Shoman KP. Sir, could you please open your
Oh, hi, sir.
Shoman sir? Shoman sir, could you please open your camera? Is it there?
Just 1 second, okay.
Yeah.
Oh, I am not.
Not in camera. No, no problem.
Thank you, sir, and hope you find my presentation useful, meaning you somewhat interesting.
Yes, 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 yes. We are trying to enter into this area, both sides, like discovery side as well as the genome sequencing, and both areas
Right.
are moving extremely fast. So through these type of talks only, we can really update.
Understand. Thank you.
Anyone else? Do you have any questions? Please go ahead.
Hello, good morning, sir. Uh,
Hi. Good morning,
Hello,
Shruti.
good morning, sir. Good morning. So, sir, my doubt is regarding the toxicity of these drugs, sir, because
Right.
so as you were saying that it only takes very less amount of time to develop these drugs, right? Or novel molecules which
Right.
can be.
Right.
So, can NLP help in predicting the toxicity of these molecules? Uh,
Yes. Yes. That's one of the application of NLP. You can actually see a lot of publications using self-attention models or LSTM-based models or even the simple regression-based models or graph-based models, where they help users to predict not only the molecule whether toxic or not, it will also tell you which group is responsible for the toxicity.
okay. Okay.
And
Thank you, sir.
one more point I want to add, you must be knowing like a carcinogenicity or genotoxicity.
Yes,
Now
sir.
FDA approves and accept machine learning model prediction whether the given compound is toxic or not. You don't need to test the compound in the mice and confirm whether if the compound is safe or safe. You can avoid that step. You can based on simple ML model, you can say that this molecule is safe.
Okay. 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 Thank you, sir.
Thank you. Thank
Hello,
you.
sir. Sir,
Yeah.
Yes,
Hi.
go ahead.
yeah, sir, I have a question actually. So suppose if I have a multi-component mixture, suppose some oil samples. So it that consisting of variety of compounds, such as like aliphatics, aromatics, or heterogeneous compound, or
Right.
like sulfur compounds,
Right.
nitrogen compounds. So which model,
Right.
which is will be best for like getting the good yield or something? Which will be, what is your suggestion? Which model is good for doing this NLP analysis?
Okay.
Yeah. That means like
Um,
multi-component mixtures.
right.
So yeah.
Right. I understand. There are actually, lot of AI-based yield predictions or compositions is happening, but it's too early to say anything. Uh, I think SciFinder has some model, and sorry, it's a Reaxis has some model to predict the yield. But there are some open source models also available that can predict the yield for a given molecule or scheme. But still, I can say we can't completely trust those models. It's too early to apply AI or ML-based models to predict the yield because these models are simply trained on the schemes, are simply trained on one certain parameters. But when it comes to the lab, there might be a number of conditions that influence the yield. Even raw material purity, temperature, or pH, and impurities, there are a number of things. So still, there is a model need to be evolve a lot to predict it's to be more accurate.
Yes, sir. Yes, thank you, sir. Sir, one more question actually regarding the
Thank you.
toxicity. You have specified that, right, sir? Because for application point of view, so we have to see the toxicity of the drugs. So that means mainly how it affects on human beings or plants or other species. So we have to see the
Right.
genotoxicity, also the cytotoxicity. So what about the
Right.
cytotoxicity aspect? So how it will be like affecting these models? So what is the comparison between cytotoxicity and genotoxicity in that direction?
So yeah, so simply the genotoxicity is nothing but of a given molecule has any groups that can actually damage our DNA, genetic material, or cytotoxicity is nothing but of whether the given molecule can kill the normal cells or is only toxic to cancer cells. So there are certain models that is FDA reviewed and approved that can predict the genotoxicity by giving a smiles. You can actually predict whether molecule is good or bad. It may safe or not. But the cytotoxicity, I think Lhasa has only one model that can predict the cytotoxicity or carcinogenicity as well.
Okay, thank you, sir. Thank you so much.
There are some open source, yeah, there are some open source models you can explore, but make sure you can trust the models.
Yeah, thank you, sir.
Dr. Navani,
Thank you.
you can ask question. Dr. Navani raised your hand. So Navani, please go ahead.
Yeah, thank you, Prati. So, thank you, Dr. Suneel, for such a nice talk. So, I have a doubt regarding your one of the case studies. So, I
Right.
think the third one, you in that flow chart, you showed that after generation, you had something like 30,000 compounds, right? And
Right.
after that, we had the filters to be applied, and then maybe reached like 4 or 5
Right.
compounds. So,
Right.
30,000 compounds generated using AI. Is it like then will it be too much time consuming with those filters itself, or uh, is
No. So basically like finally what you're expecting, the LSTM or any model, it can generate n number of molecules that you want. And applying filters is not that time consuming, except talking will take a time. But after once you generate a 30,000 compounds or 1,000 compounds, you can simply calculate diversity and you can narrow it down some number and followed by toxicity and followed by drug-like properties or followed by pain alerts. So you can actually reduce that number drastically after applying these filters. And followed by docking will further reduce into the number into the maybe 2 digits or maybe hundreds or 200s, which
so
you can
these fil
handle and assess manually. Yeah.
yeah, these filters are applied in series, right? So you choose something at first, and then you after the first filter is
Right.
applied, you do it in such a way. Okay. So one more.
So either you can do automated. You can tell the system to, okay, after generating compounds, you apply these filters and initiate the docking studies and what are the molecule that that passes the drug-like rule or paints alert or toxic alerts and docking with a score of maybe -9, it automatically sort for you and will give you the molecules of your interest.
Okay. So one more doubt regarding what
Yeah.
you had taught. Uh, so you had told about some cons of reinforcement learning. Can you little bit elaborate on that when using this?
Okay, so reinforcement learning is nothing but of a, I can say, very simpler example. When kid is crying, you're actually giving a chocolate to, make him happy, right? So that's nothing but of a reinforcement learning. In a similar way, we are applying reinforcement learning while generating a molecules. Uh, Whenever the molecules are generated, you know, imagine like a molecular weight, like what we discussed. If the generated molecules are molecule falling within the molecular weight of our choice, so the reinforcement learning will give you the positive awards. If not, negative awards that will discard it during the generation process.
Yeah, but I think in your what you suggested as a con was that there is a large chance of these compounds being discarded when you are using reinforcement learning, right?
Right. So that's
Potential.
what my no, that's what my understanding. Like imagine you applied a filter of 300 to 400 molecular weight, uh, but there is a nice molecule has a molecular weight of 401, so you might miss. So when you are applying too many filters, you ended up sometimes filtering a noise, sometimes filtering a good commutates as well. So that's up to you whether are you okay to rule out those compounds.
Okay, understood. Thank you for the
Yeah.
clarity.
Now in a given competition power, I think you can easily handle the number of compounds that is generated.
Yeah. Okay. Thank you for the
Thank
clarification.
you.
Thank you.
Okay, Dr.
Thank you.
Sunil, now on the screen, Dr. Soman KP is there, and he is our dean. Just before few
Hi, sir.
minutes, you talked to him. Okay.
Hi, sir. Nice to
Another
connect
raise,
with you, sir. Thank you for your time.
and we have another hands on. Shubhasri, please go on.
Hello, sir. Very nice talk. Thank you. I have one question.
Thank you,
So,
Subhasri.
yes, sir. So, we have a lot of large language models
Right.
which have trained ChatGPT like models. So, are there any chemical language models that we can leverage for this purpose?
Yeah, there are some interesting groups in Canada and US. They're already started working on the large language models, but still we are not there yet. It's a long way to go because of the data set that we are going to train. But if you want to explore, there are data sets that are trained on the chemical, that are trained on the uh, given chemical data sets. Yes, but we can't completely trust those results or whatever the analysis that generates.
Okay, sir. Yes, sir. So one other doubt,
Thank you so much. Yeah.
instead of generating molecules, can we
Right.
leverage AI in screening using the traditional docking methods because they are so robust? And can we do that?
Sorry, Subhasri, can you come again? There is a lot of
Yes.
echo. I can't hear.
I was saying that instead of using generative AI to come up with new molecules, can we use AI in screening, drug screening, using docking itself? Because docking softwares are very robust. So, how can we use AI to speed up the screening process?
Sure. Dr. Sunil, please unmute yourself.
Uh,
Can you do unmute, Dr. Sunil? Yeah.
Okay. Yeah. So to answer your question, Subhasri, you don't need always a generative AI or you don't need docking always. to generate ideas or to identify best compound. There are a number of methods. You can generate a molecules using pharmacophore or docking, scaffolding, bioisoster replacements, or shape-based filtering. Always there are a number of combinations are available based on the given situation, given target, given data set. You can actually pick the right approach for your study. Always docking is not a solution. Always generative AI is not solution. So the user has to understand the pros, limitations of the approach, and uh, take the right strategy for the study. Like imagine you have a 20 billion compounds. Can you dock 20 billion? No. So you can use some ML model to narrow down that 20 million, make it to maybe 1 million or half million, and then dock it. So there is actually based on the given objective, you can smartly choose which approach is better. And coming to your second question, if you want to use AI in the virtual screening, yes, there are strategies called active learning. There are strategies called deep docking. You can use those techniques that uses the docking. But again, docking is always, the time consuming. On an average, one molecule takes 6 minutes. When you are applying deep docking, it might take more time. If you are okay to spare the time, yes, you can try those methods.
Thank you so much, sir.
Thank you, Sudarshan.
Audience, any questions? Suneel, thank you for a nice talk once more, and thank you for joining us at the late night because it is already 12:30 a.m. in your side. And all audience, thank you to make the session too interactive. Thank you, Suneel.
Thank you,
sir.
everyone.
Soman
First of
sir,
all,
thank you. You are here. Yeah, Suneel, carry on.
Thank you, everyone, for the opportunity. Thank you, Pratiti, for kind invitation, and thank you, everyone at Amrita University, for inviting me and hosting me. And thank you so much, sir, for your time and sparing me for my lecture. And thank you, all the audience. I'm not sure whether I addressed your questions or responded correctly or not. If you have any questions, feel free to reach me on LinkedIn, or I'm happy to address. My email ID is available with Pratiti. I'm happy to always support. Thank you.
Thank you. Thank you.
So we are closing that meeting. Thank you, Soman sir.
Thank you.
So we are closing the meeting, the seminar. Thank you all.
Thank you.
Bye. Have a nice day.
Thank you. Bye.